Great, so I'll get started. Thank you everyone for attending today. My name is Mayukh Chakrabarti, and I'm a graduate student in the lab of Dr. Amzel at the Johns Hopkins University School of Medicine. Today I'll be telling you about some of my recent work investigating dynamic allostery and PI3K alpha using molecular dynamic simulations. So I'd like to start by first providing a brief introduction about PI3K, which stands for phosphatidylinositol 3 kinase. So PI3Ks are generally divided into three classes on the basis of subunit composition and substrate specificity. Within the class 1A is a further subdivision into class 1A and 1B on the basis of regulatory subunit. Today I'll be telling you specifically about PI3K alpha, which is, it consists of a P110 alpha catalytic subunit and a P85 regulatory subunit. And PI3K alpha utilizes ATP hydrolysis to catalyze phosphoryl transfer at the 3' hydroxyl position of phosphatidylinositol 4,5-bisphosphate, or PIP2, giving rise to phosphatidylinositol 3,4,5 triphosphate, or PIP3. And this regulation of PIP3 to PIP2 can be modulated by this phosphotensin and homolog enzyme, which is P10. So in a cellular context, PI3Ks function downstream of receptor tyrosine kinases. And as you can see in this diagram, upon substrate-bound RTK activation, PI3K is recruited through the binding of the P85 regulatory subunit with phosphorylated tyrosine residues. And in this diagram is shown the P85 interacting with the phosphorylated adapter protein IRS. Let me see if I can get a pointer here. PI3K then produces PIP3, which acts as a scaffold for a number of downstream proteins that contain plexion homology domains such as AKT and PDK. AKT can then participate in a number of signaling pathways that regulate multiple cellular phenotypes, including cell growth, transformation, survival, and apoptosis. So as I mentioned, PI3K consists of a P110 catalytic subunit and P85 regulatory subunit. Within the P110 catalytic subunit is an adapter binding domain that interacts with the inter-SH2 or ISH2 domain of the P85 subunit. It also consists of a RAS binding domain that binds the RAS protein, a C2 domain that interacts with the plasma membrane, a helical domain whose function is not known, and a kinase domain which contains the catalytic site of the enzyme. Within the P85 subunit is an SH3 domain, a domain that contains homology to GTPase activating protein, hence termed a gap domain, and 3 SH2 domains, the NSH2 domain, which binds phosphotyrosine peptides, and an inter-SH2 domain and a CSH2 domain. So the construct that's shown here, or the structure that that's shown here, was solved by our lab and represents a structure that is both necessary and sufficient for observing the activity of this enzyme. So focusing more closely on the kinase domain, we can see that the kinase domain consists of an N-terminal and C-terminal lobe and shares structural similarity with other known protein kinases, including the presence of residues such as the DFG motif. And today I'll be focusing specifically on the activation loop, which I'd like to bring your attention to here. And these residues are in proximity to both the ATP and the lipid substrate that participate in the catalytic mechanism of this enzyme. So work by Burke and colleagues regarding PI3K have shown that that mutations can actually allosterically modulate motions in distal domains of the protein. And this was done using hydrogen deuterium exchange mass spectrometry, which looks specifically at the exchange of backbone amide hydrogens. And as you can see from these figures, mutations, for instance, at N345K in the C2 domain and E545K in the helical domain can cause changes in the dynamics in other regions of the protein and also in the P85 subunit. Moreover, we can see that upon binding a phosphotyrosine peptide effector in both wild type and mutated PI3K, we can again observe perturbations in the domain fluctuations in distal regions of the protein. So what this tells us is that PI3K can be allosterically regulated. And what we also know is that this type of allosteric regulation can also increase the catalytic activity of the enzyme by several fold. We know from our understanding of protein kinases that active site residues mediate the catalytic phosphate transfer. But the puzzling observation is that even though we have more than 50 crystal structures of PI3K currently in the PDB, none of them actually capture this the catalytically competent state of the enzyme. That is what the enzyme looks like when the catalysis is occurring. And so the question then logically arises is, if we could somehow modulate the dynamics of the activation loop or residues in the active site, would that play a role in populating this catalytically competent conformation of the enzyme? So to understand this question, we first turned to work that was performed in our lab investigating the kinetics of this enzyme. And specifically, we performed mutations in active site residues as well as residues that are proximal to the active site. And what we find is that specifically mutations in histidine 917 and histidine 936 can abrogate substrate binding as well as enzyme activity. And on this basis, for some of my simulations, I applied harmonic restraints that maintain the proximity of these residues to the ATP and the lipid substrates. And I set these restraints at 3.5 angstroms and a force constant of 2 kcal per mole per angstrom squared. So in this work today, I'll be telling you about several simulations that I performed. One is a set of simulations that I performed on the wild type system. Another one is a set of simulations I performed in which the NSH2 domain is removed. And this set of conditions is meant to mimic the activated state of the enzyme. I also performed simulations in which I bound the phosphotyrosine peptide, as shown in this video. And I applied here conditions with distance restraints and without distance restraints. So what I find is that, in fact, by displacing the NSH2 domain, or in this case, not having the NSH2 domain in my simulations, is able to actually allosterically perturb the dynamics of the activation loop. And what we can see here is the RMSF profile of systems in which the NSH2 domain is not present in red, and in which the NSH2 domain is present in black. And as you can see here, these are the residues of the activation loop. And you can see that the RMSF profile here it differs significantly in this region. Moreover, we find that the removal of the NSH2 domain actually influences interactions of active site residues with the lipid substrate, as well as the interaction of the ATP with the lipid substrate. And as you can see in these tables, in the presence of the NSH2 domain, these interactions are much more frequent than in the absence of the NSH2 domain. However, what we observe is that it's only in this case when the NSH2 domain is not present and these interactions are less frequent that we actually observe conformations that are compatible with a catalytically competent state. And so here I'm showing an instance from my simulations in which the epsilon nitrogen of histidine 936 is within 4.1 angstroms of the O3 of the PIP2 lipid substrate, and the O3 is within 4.9 angstroms of the gamma phosphate of ATP. Here's another instance from my simulations in which epsilon nitrogen of histidine 940 is within 3.4 angstroms of the O3 of the PIP2 substrate, and O3 is within 3.6 angstroms of the gamma phosphate of ATP. So I also find that, in fact, that the removal of the NSH2 domain can alter the conformational ensemble of the activation loop. And what I'm showing here are clustering results from wild-type PI3K and PI3K in the absence of the NSH2 domain. So as you can see from the top row and comparing the top row and the bottom row, you can see that the conformational ensemble of the activation loop is actually altered by removal of the NSH2 domain. I next looked at binding of phosphotyrosine peptide effector, and I found that it too can also allosterically perturb the dynamics of the activation loop. So as you can see here again, in simulations in which the phosphotyrosine peptide is present, the fluctuations of the activation loop are decreased significantly relative to simulations in which phosphotyrosine peptide is not bound. I also looked at correlated motions between the NSH2 domain and the activation loop. And what I'm showing here is a correlation plot, and it's a heat map. So it's essentially the Pearson correlation, +1 being perfectly positively correlated, -1 being perfectly anti-correlated. And as you can see here, in the presence of the phosphotyrosine peptide effector, the correlated motions between the residues of the NSH2 domain, shown here in the x-axis, and the activation loop in the y-axis, are strongly perturbed relative to the cases in which phosphotyrosine peptide is not bound. Moreover, when I apply active site distance restraints in the presence of this phosphotyrosine peptide effector, I again see changes in the correlated motions between the NSH2 domain and the residues of the activation loop. And strikingly, what I observed that in both of these perturbations, that is a removal of the NSH2 domain and binding of phosphotyrosine peptide effector, we can see a similar conformational ensemble adopted by the activation loop residues. And this is again shown here in the clustering results. And you can see here in the top the residues of the activation loop in the cases in which NSH2 is not bound, and in this case, when phosphotyrosine peptide is bound. And however, this result, the convergence of this result, is not due to the simulation sampling the same conformational space. So what I'm showing here is a covariance matrix of the displacements of both of these backbone displacements of both of these simulations. And these are projected on the first two principal components. And as you can see from this plot, the two sets of simulations don't overlap in this PC space, indicating that they're actually sampling different regions of conformational space, yet the activation loop ensemble is converging to a similar result. And so to explain this, these observations, we propose that PI3K actually has this mechanism of dynamic allostery. And dynamic allostery itself as an idea is not new. It was analytically shown by Cooper and Dryden in 1984, and has since been demonstrated experimentally in multiple systems. So what we can see here is based on this idea, in wild-type PI3K, the population of the catalytically competent state is so low that we in fact don't really see that active conformation or catalytically competent conformation even in multiple crystal structures of the enzyme. However, when we allosterically perturb PI3K either through removal of the NSH2 domain or binding of phosphotyrosine peptide, the population of the catalytically competent state is increased without significantly altering the ground state conformational ensemble. And thereby, this catalytically competent conformation has a higher representation, and we see it with a higher probability in the samples, in the states of the enzyme. And this allows us to observe such catalytically competent conformations on the timescales of our simulations. So to summarize, what I've shown here is that we can indeed alter the conformational ensemble of the activation loop through displacement of the NSH2 domain, and this actually increases the population of active conformations and lipid substrate interactions that may lead to catalysis. Similarly, I've shown that in binding of phosphotyrosine peptide effector can also allosterically modulate this conformational ensemble in a way that can lead to catalytically competent states. So I'd like to finish by acknowledging my advisor, Dr. Mario Amzel, Dr. Sandra Gabelli, who was involved in solving the structures of this enzyme, and as well as current and former members of the Amzel lab that were involved in kinetically characterizing this enzyme. I'd also like to acknowledge funding sources as well as the computational facilities that I use to perform this work. And finally, I'd like to thank the SBGrid organizers for providing me a platform to present this work today. And thank you all for listening. I'll be glad to take questions.
All right, great. Thanks, Mayukh. Again, if folks have questions, feel free to send them to me or Carol in the chat. One question that, for folks that aren't familiar with molecular dynamics as much as you are, what is RMSF measuring?
So RMSF is a root mean squared fluctuation. So it looks at the fluctuations of residues about an average. So okay. If there's a fluctuation, so if there's an increase in fluctuations, it means that the residues are deviating from their positions at a certain distance relative to the average conformation. So there's an increase in fluctuations of these residues. So so essentially the residues here are exploring conformations which are deviating by a certain distance from the conformations that they would ordinarily have in a time averaged manner.
And does it make a difference if that's Cartesian coordinates or internal coordinates for that? Or
I mean, the results should still be the same. I mean, because it's measuring a deviation. So the deviation shouldn't depend on what system is used to measure the deviation.
Great.
But in this case, it's usually shown as it's represented in a Cartesian manner.
Thank you. Another question: how does PIP2 affect these simulations?
So PIP2 is the original substrate, right? So this is what's getting phosphorylated at the 3' hydroxyl to lead to the production of PIP3. So PIP2 is one of the two substrates in the reaction. One is ATP and the other is PIP2. So these two substrates need to be present in order for the reaction to occur.
Okay, great, thank you. Could you say a little bit about, so you've got a great story and a great hypothesis for how the dynamic allostery is working.
Mm-hmm.
Can you say anything about what you think for how you go from the simulation to confirm, like to try to get experimental confirmation on that?
Yeah, so that's the difficulty. So this enzyme, what I didn't mention here, so P110, so the 110 actually is the weight of this subunit in kilodaltons. So this is 110 kilodaltons catalytic subunit, 85 kilodaltons regulatory subunit. So altogether, this is about 200 kilodaltons, and it's two subunits. And so it's a very large system. And if you try thinking about even doing NMR or any other method, trying to probe these kinds of dynamic motions in a large protein like this is very, very difficult, which is one of the reasons that I had to try investigating this system computationally. It's very difficult to look at such a system, such a large system, and try to understand exactly what's happening. And this is a way that we might get closer to that. And so I would be glad to hear if people know how to do something experimentally on such a large system to look at intersubunit dynamics, and that would be really great.
And my sense, and you'd know better than me, is that this is a large complex for NMR, and this is also a relatively large complex for simulations too. Right,
right. It's also large for simulations. In this, wait, I showed here the number of amino acids here is around 1,000 amino acids here and another 300 or 400 amino acids here. So this is relatively large. Even what I'm simulating is around 1,600 or so amino acids. So it's a relatively large system, and I'm also simulating it. Trying to look at the dynamics of such a large system is difficult.
That's what makes it interesting.
Yeah, and that's what makes it interesting is even the fact that I can observe these types of changes and fluctuations is very interesting.
Yeah.
So, yeah.
Thank you again.